walk you through how to do malaria cell detection using a couple of CNN models. So the data set we have is around 300 meg with two type of labels. We have parasitic and uninfected labels. What I'm going to do is use some of the template projects from BrainCradle and quickly run up a few models and see how my performance goes. So what I'm going to do is I'm going to go to I have a template project with quite a few models, and all I'm going to do is clone them from this project on into my malaria project. So there you go, it's cloned. And the only few changes I need to do is if I go into data input, I would need to tag it to my data set, and I'll change my split so that all right, my model input looks fine. Go to the output. Since I know I have two labels, I'm going to change that to 2. My training machine looks, basic GPU looks good, batch size looks good, epochs, my loss looks good, the metrics to captures accuracy is fine with me. And let's look at optimizers. The default is Adam, that looks good too. So I'm all set, and I click the training button, and the training request has been submitted, and I see my status. So while this is training, let me go in and actually go back to the model template. So this was a 2-block CNN. Let me clone the 3-block CNN. And let me just quickly look through the model. Everything looks good. It is a 3-block CNN. I'm going to attach my data set to this, change the training validation split. The make sure my input size is the same. Go down for output classifier, pick 2 since we have 2 labels, and I'm going to keep all my hyperparameters the same. Let it run on a basic GPU. Check optimizer, Adam's good, and say train. So this is my second model being trained. Next, I'll go back to my model templates, and let's try a VGG16 with weights with the frozen base and perform some transfer learning. So clone that project model over, and I'll do the same thing, just attach my data source, and let me keep everything the same. And for the model output, set it to 2. I will keep everything the same, and the default what has been selected, and click train. So this is the VGG16 with the frozen base is also started training, and I'll do one more. I will pick the VGG19, which is also with pre-trained weights and frozen base. Clone that over. Okay, so I have the VGG19. You can see the model with a bunch of layers. Go back, pick my data set, and change the split to 70-10-20. Okay, and go to my output and change it to 2 since we have 2 labels in our data set. And in this one, I'm going to change the batch size to 32, and everything else I'll remain the same, and say train. So basically what I have is I've quickly cloned 4 models into my new data set, and I have these four models being trained. I can see the status. um, Obviously these models are not going to train like in a couple of minutes. They're going to take some time, but what I'll do is I'll come back later and then we can see the progress of training and the results of training.